Hello, this is your by request April Fool's Day vodcasted lecture, and if you are watching this on the day of the regularly scheduled lecture, you are hopefully in lecture participating in the activities that we're going to do that are very active and get you jazzed on proceeding forward with your poster. Maybe that poster is the one that you're doing with your group members, or maybe instead you opted to work on your individual metabolism and gene expression poster, but either way we are doing stuff in lecture right now, so go to lecture. So that being said, please do also finish your participation in the Follow Your Gut discussion. I'd really love to be able to get to looking at that hopefully on Sunday, but as soon as possible so that we can move along with that and have one more discussion before we end the semester. I really do need the poster hypotheses to be finalized, and hopefully if you haven't gotten that done, we will be getting that done in class today. So please do think about getting those submitted to me in their finalized form, because ideally I would like to receive your abstract sometime between April 6th and the 13th, showing me that basically your project is done and that all you need to do at this point is to make the poster and create a very user-friendly kind of interactive form that will allow you to talk about the project that you've done and really effectively communicate the project that you've done. So homework 7 is our next homework, and then looking into the future, we are going to be looking for exam number 3 on Tuesday, April the 12th. So that'll be our third and last kind of, it's not traditional because you're allowed to use your poster, but it's the last one that you need to come to the lab to take. And the final exam, of course, will be fully take-home, open note, open resource, and all of those fun things that accompany a really sexy-minded exam like the final exam. Okay, I want to jump in because when we ended off last time, we were thinking about tRNAs, and this morning I had the most awesome visit with Brad. He came in with a set of adapters that he said, well, I don't understand this whole tRNA serving as an adapter idea, and so he's promised me he's going to make a short little video clip showing the adapters that he had for phone lines, I believe, and for sound transmission, but he showed me how one adapter went with the next, and I said, okay, you could make an analogy that this is like a tRNA. So when we left off, we were thinking about Francis Crick terming tRNAs adapters. So I have an analogy that I've cooked up, Brad. It's not probably as good as yours, but if you take this adapter that I have for my phone, this is an adapter that will take my phone, so I can plug in my phone here, and it will allow it to plug into a projector. So if we think about the phone as being the amino acid, so like lysine attached at the acceptor stem of the tRNA is the amino acid like my phone, this would be like the tRNA. And then the recognition of the projector would be like the mRNA. So the mRNA would be the projector, this adapter would be like the tRNA, and my phone would be like the amino acid. So that's what we see those tRNA doing, is recognizing the codon and adapting it to the amino acid that it carries. One thing that I didn't note is that the addition of the lysine amino acid onto the tRNA is an enzyme catalyzed process. So you noticed how I took my phone and I plugged it into the supposed tRNA there, right? That is enzyme catalyzed. So there's an enzyme called an aminoacyl-Trna synthase that actually does, I was the enzyme there, right? Plugging the phone into the adapter, the enzyme aminoacyl-Trna synthetase would actually be plugging the, adding the lysine onto the acceptor stem. The 3' hydroxyl is the binding site of the amino acid lysine. So of course that means that this is the 3' end of the tRNA and this is the 5' end. And of course that was important to our discussion of the anti-parallel nature of the anticodon and the codon. I also briefly mentioned that tRNAs don't really look like this flat on a page clover leaf structure, and instead they gain tertiary structure. This picture gives you an idea of what a tRNA would look like when it was actually free to form its 3-dimensional structure. Notice that the acceptor stem where the amino acid would bind, the 3' hydroxyl, is up here, whereas the anticodon that recognizes the codon is here on this side of the sort of L-shaped tRNA molecule. We recognize then that tRNAs play this extremely important role in recognition of the mRNA codons, And we also mentioned that tRNAs are the root of degeneracy. So remember talking about wobble, or the loosey-goosiness that takes place at the most 3' side of the codon, which is the most 5' side of the tRNA. So remembering then that there is a wobble there, that means that there are not 64 tRNAs. There are less. There are around 40-some tRNAs that recognize all of the 61 codons. Remember, some are stop codons, so those don't have a corresponding tRNA. But then the rest are recognized by the tRNAs that can have a little bit of wobble or loosey-goosiness in their most 5' anticodon position. Now, we need to do an example of this because it's sort of hard. It's easier once you can actually visualize this. So I actually want to draw an example of where we would see the wobble effect taking place. Let's assume that it's a GCU anticodon. So we know that up here is up above is the 5' end, this is the 3' end where the amino acid is attached. We know that this anticodon would then recognize the codon and the mRNA, and we know that it could pair, as we might expect. A G would pair with a C would pair with a G, and a U would pair with an A. So we could recognize that that codon would be one possible codon that this tRNA could recognize. And if you quickly look this up in your amino acid usage table, and we can go ahead and do that. I'm going to go ahead and bring up the amino acid usage table, the codon usage. GCA is what we're trying to look up. So GCA, or sorry, CG. I'm having my own translational issues. So CGA, so CGA, right? That's arginine. Now, when we look at degeneracy in the code, we might start to guess that, hey, maybe the wobble in this tRNA would enable this U to also recognize a G, and that is absolutely true. So it is possible that this tRNA might additionally recognize not just codon CGA, but it could potentially also recognize the codon. So we could say in an alternate possibility, this could actually be the codon CGG, so CGG also encoding for arginine. That is the wobble effect, right? That U could either pair with an A or it could pair with a G in that position. We know no matter what, way up here on the 3' end, on the acceptor stem, no matter what, that amino acid is going to be arginine that this carries. Beautiful. So that hopefully exemplifies the wobble effect. And I can give one other example that we can write down in the notes. And my suggestion is to go ahead and draw it the way that I've just drawn this for the CGA and CGG examples. You could also draw the example that we have written in the notes here, which is the fact that CGG can recognize both GGU and GGC. So you could say that was another example of wobble. So go ahead and look that up and see if you can tell me what amino acid would be recognized in both cases, no matter whether it's GGU or GGC. Beautiful. So now we need to move our discussion to the 3 phases of translation: initiation, elongation, and termination. And what's really interesting is that in bacteria, we can actually see that translation will begin before transcription terminates. That is weird to think about. Why is that true in bacteria, but it wouldn't be true in eukaryotes? That's what we need to ponder. As we look at initiation of translation in this really cheesy drawing, which by no means is to scale, notice that we've got the RNA polymerase enzyme. It's during its elongation phase of transcription, And recognize that even while elongation is still taking place and the mRNA is building 5' to 3', adding new nucleotides, new NTPs to that open 3' hydroxyl, at that exact same time, the ribosome has recognized its site to bind and has begun reading the mRNA and building the polypeptide chain simultaneously. So this can actually occur at the exact same time in a bacterial cell. So hopefully you thought about why this wouldn't be true in eukaryotes, and the compartmentation provides a preventative to this, because we know that translation is occurring in the cytosol, whereas transcription was occurring in the nucleus, and so that's why we don't see these happening simultaneously in eukaryotic cells. That being said, let's look a little bit more at the nuances of the initiation process. And let me tell you, there are a lot of nuances because when an mRNA is recognized and the process of translation is about to begin, it's the 30S subunit that first recognizes a region in the non-translated leader sequence of the mRNA. And that's a very special region. This is called the Schindel-Garno sequence. So I actually want to do a little bit more up close and personal drawing to show you what's going on here. If we draw a really huge depiction of the 30S subunit, inside of that 30S subunit, we know there is the 16S ribosomal RNA. And this 16S ribosomal RNA is going to be anti-parallel to the mRNA. And on this 16S ribosomal RNA, there is going to be a region, and I'm going to actually put the sequence into this region. It is a very pyrimidine-rich sequence. So say something like UCCU, very rich in pyrimidines. And so then on the mRNA, which I'll go ahead and show in red because it's shown in red in our picture there, here's the mRNA, and it's going to be recognized by this sequence. And so here we're going to have something like AGGA on the mRNA that's going to pair with that UCCU. So that sequence right there is this sequence here. It is the Schindel-Garno sequence. The Schindel-Garno sequence is going to pair with the 16S ribosomal RNA. And that's why the 30S ribosomal subunit recognizes the mRNA and says, whoa, hey, everybody stop right here. This is where we begin. Now, the 30S subunit is not the only thing that will be called to that initiation site. Additionally, there are going to be some initiation factors that play a role. And also remember that once this mRNA is all aligned with the 30S ribosomal subunit, somewhere downstream of the Schindel-Garno, of course we know we're going to find the AUG start codon. So somewhere downstream there will be the spot that begins the pairing of tRNAs with amino acids on their acceptor stem and begins that process of converting the mRNA code into the polypeptide. And so this is what we're showing here. Right there is the AUG start codon. And we know that the tRNA that carries an FMET, an N-formal methionine that we drew last time, is going to be bound to that acceptor stem. So this is the start codon and the beginning amino acid, the N-formal methionine. But notice that there are these interesting initiation factors. This one right here is very famous. It's called IF2, or initiation factor 2. Initiation factor 2 is going to make certain that the right tRNA, the right activated tRNA with the FMET is bound to the AUG start codon. Notice too that IF2 has a GTP Woohoo! Energy, right? We know that just like transcription was highly endergonic, requiring a lot of energy, so too will be translation. So with every initiation event, this GTP is going to get hydrolyzed. So this is a pent-up reserve of energy there. There's also a couple other initiation factors involved. IF3. Guess what IF3 does? IF3 prevents premature association of the 30S ribosomal subunit with the 50S, so it stays unassociated until IF3 leaves. IF1 is less well understood, though it seems to be that it coordinates the activity of IF2 and IF3. So this being said, this is what we call the initiation complex. So this initiation complex is extremely important in kicking off the process of transcription, of getting everything going. So it all comes together at the start codon, and this initiation complex with the FMET and the initiation factors all come together at this site. So this is most of initiation, but we're missing something pretty dang important, right? We are most certainly missing the 50S ribosomal subunit. So at this point, recognize that the IF2 is going to hydrolyze GTP, the initiation factors are going to get the heck out of Dodge, and the 50S ribosomal subunit is going to swoosh in. So this is going to end the initiation phase of translation, and we're all ready to go. Everything is poised to run right into the elongation phase, where now more tRNAs can bind, recognize subsequent codons, and we can get formation of peptide bonds and the elongation of that polypeptide chain. Before we move into elongation and get really in the depth of that process, we should practice and see if we make decent ribosomes at all. So let's check it out and see what's going on in this question. Here we have an mRNA sequence, and in this particular case, I've shown the Schindel-Garno sequence as just a black box. But of course, all of you recognize and understand that rather than a black box, that would be a very purine-rich sequence. For example, maybe it would be a GGA, as we saw in the last example. 16S ribosomal RNA will be called too close to that sequence so that the 30S ribosomal subunit will associate with that region, allowing everything to align in such a way that the N-formyl methionine can bind with the start codon. So in order to make really good ribosomes, then, we have to scan down from the Schindel-Garno sequence to see where the first AUG start codon is in order to set the reading frame. So that's always going to be downstream of the Schindel-Garno sequence, and we'll find the first AUG knowing that the N-formyl methionine tRNA FMET, right? The tRNA that's destined to carry N-formyl methionine and does. So it will align then with its anticodon arm binding with that AUG. So we know that the first amino acid will be the FMET. And we know that this, of course, is the amino terminus of the polypeptide chain. And we know that as the ribosome continues reading, as we head into elongation, our GUC can be looked up on the codon usage table. So we need to look up GUC, valine. So our highly hydrophobic valine will be next. So this will be val. Then we can go on. Oh, hey, look, AUG. So AUG only serves as the start codon once. After that, it quite simply encodes for methionine. So the second time the AUG is encountered, we just incorporate methionine. I suggest you keep on going with this. Keep on reading down codon at a time and adding amino acids a little bit at a time. Beautiful. Now that we understand how a ribosome is going to go about this, let's go into the nuanced details of all of the sites that we would find in a ribosome and how they accommodate the process of elongation. So once initiation has taken place and the 50S ribosomal subunit has swooped in and has bound, we can pick out some really pertinent spots inside of the ribosome. These are actually given specific names. So it's like landmark sites inside of the ribosome, and each one of them is named. These are called the EPA sites. So the first of these sites is the E site, stands for exit. The second is the P site, stands for peptidyl, and the third is the A site, standing for aminoacyl. So altogether, the EPA, ironically, are the sites that we see inside of the ribosome. Now, let's see if we can kind of picture, after initiation has taken place, where do our other players fit in? So remember that our aminoacyl-Trna that carries the FMet, or the informal methionine, is already bound. And in fact, it's going to be bound right here. So the tRNA, and this is going to be just a terrible, atrocious drawing, but we know that the amino acid there is the FMet. The FMet is already inside of one of these sites. And if you look carefully, you'll notice that the site that it's inside of is the P site. So after initiation, when the 50S ribosomal subunit adds, the P site is immediately occupied. It's occupied by the N-formal methionine tRNA FMet, the FMet carrying tRNA. That leaves open the aminoacyl site, which means that, so let's write in our mRNA player here so that everybody recognizes the mRNA is kind of fed through here, where the 16S ribosomal subunit, or the 16S ribosomal RNA, binds with the Shindell-Garno sequence right here at the mRNA binding site. And then, of course, this will be, and I'll just use some colors to dictate this, this codon right here will be the AUG, and then the next codon, of course, will be the second codon, and that sequence will be, you know, whatever 3 nucleotide codon happens there, which will dictate which tRNA will then bind at the aminoacyl site. So the second tRNA with the right anticodon to adapt to the next amino acid will swoosh in. Now, you might have guessed that that's going to require energy, and it does. In fact, it requires quite a lot of energy, and there's another factor that helps to allow the tRNA to clip into this A site. We call it EF2. Let me jot that down because it's not the number 2. It's the elongation factor, TU, and it serves, I like to think of it as being like the ski boot of the next aminoacyl tRNA, because it allows the aminoacyl tRNA to clip in to the A site. So it plays a large role in that, and more ATP or ATP equivalents, GTP if you will be hydrolyzed to power that. So it's a highly endergonic process. So now that we kind of see that happening, let's move our view into elongation. So the second tRNA will bind at the A site. So we could say within elongation, we have 3 stages. So elongation can be broken down into 3 separate stages. Aminoacyl tRNA binding, April Fools. Okay, so aminoacyl-Trna binding is the first step. 2, notice what's got to happen here next. We've now got a tRNA with its amino acid bound in the P site, which of course we know to be FMet. And we've got a tRNA with its amino acid bound in the A site. Well, what has to happen next is that a peptide bond needs to form between those 2 amino acids. We know that the 23S rRNA inside of the 50S ribosomal subunit is what's going to catalyze that. So step number 2 is peptide bond formation. We actually sometimes give that a sexy-minded lingo term. We call it transpeptidation. transpeptidation. And then the third step is that now that this has happened, the ribosome has to move down the mRNA by one codon. So picture it. The tRNA in the P site then gets pushed to the E site. The tRNA in the A site then gets pushed to the P site. So the tRNA that is in the E site makes its exit, right out of the E site, exit site. It leaves behind its FMET amino acid. This also leads to an open and empty A site. So peptide bond formation is followed by translocation, movement of the ribosome down the mRNA. So translocation is next. So if you can read my writing, which seems unlikely at this moment, you'll realize that there are aminoacyl-Trna binding is step 1, transpeptidation is step 2, and translocation is step 3. Let's move along and look at those happening. So there's the covalent bond formation. We know that's a peptide bond. Feel free to label it that way. That's transpeptidation. And then we have translocation, movement of the ribosome down the mRNA, pushing the N-formal methionine tRNA FMET into the E site. It leaves behind its FMET, and the tRNA itself takes off. And then it opens up the A site. So the A site is now ready for binding of the next aminoacyl-Trna. It's vacated, and so the third codon is now read and adapted to the third amino acid. And then the whole process keeps on continuing throughout this process of elongation. So we get a longer and longer polypeptide chain as the mRNA is read one codon at a time. Interestingly enough, just as we saw with transcription, translation can occur by more than one ribosome at a time. So once the Schindel-Garno sequence and this upstream non-translated region, the leader sequence, is cleared by the ribosome, another ribosome can recognize it and start reading that mRNA. We actually give this a name. We call it a polyribosome or polysome. And you could draw that out and think about what it would look like to have one mRNA being read by many ribosomes all at one time. And you're like, oh yeah, it would look like a polyribosome. Lots of them reading the same mRNA. So eventually what's going to happen is that at some point the A site is no longer going to have a readable codon in it. It'll have now a stop codon. So there will be no tRNA that will recognize that. And instead we have termination factors that will help in the process of ending translation. So we reach a stop codon. And then we're going to have transcription or translation termination. Release factors are these termination factors that help free the polypeptide chain and stop the entire process of translation. The ribosome will then fall off and it will separate out again into its 50S and its 30S subunits, and the whole thing can happen again. Now I want to show you a couple of models that my students have built in the past. I think these are pretty cool. This one simply uses styrofoam that this student has cut out, and you can see that it shows the codons with beads. This, of course, is the mRNA. Now, it could be made even better to label this, the 5' end of the mRNA and the 3' end, as the ribosome is reading down from 5' to 3'. You can see the sites in the ribosome, so this would be right here would be the P site, and we would assume that the next step to happen here would be tRNA, aminoacyl-Trna binding, where the tRNA with its amino acid would bind and recognize this codon in the A site. The E site is where, at this point, the tRNA has vacated, so they're showing in this model that it has left. And you can look at this, and I challenge you to do so. Look at this amino acid is isoleucine, and speculate as to what possible codons it could be binding with right there. This other model is really artsy. They've actually carved out wood, and this one's kind of cool because, so if we look at this in the right orientation, where the 5' end of the mRNA would be here, this is the region where the Schindel-Garno sequence would be found, and then reading down 5' to 3'. This is the aminoacyl-Trna bound in the P site and the aminoacyl-Trna bound in the A site. So at this point, we assume that the next step needs to be transpeptidation, where this amino acid would be linked to this amino acid, right? That would be the next step that would want to occur here before then translocation of the ribosome could occur. Beautiful models, love them. Extra credit, models are always extra credit. Okay, beautiful. We need to finish this off then by simply saying that after termination of translation, there are some modifications that take place. So remember that sometimes a protein is destined to be exported or secreted to be sent to the Sec apparatus, in which case it would need to have that signal sequence at the amino terminus that's added to allow it to be targeted for the Sec machinery and exported. Sometimes a chaperone protein may recognize the polypeptide chain. Maybe this polypeptide chain needs some assistance in folding, and so it will recognize that it will provide a cavern in which it can safely fold. We also will recognize something interesting happens. Remember that, of course, in all of our amino acid sequences, F-met is always the first amino acid. However, if you look at all proteins, they don't all start with F-met, and that's because post-translationally a region of the amino terminus is clipped off. So at least 1 to 3 amino acids on the amino terminal are always removed, and that always includes that F-met. So that's interesting to think about. Challenge question shown below is a prokaryotic mRNA transcript and the polypeptide that results as this transcript is translated. And what I want to know is which tRNA will bind next to the A site. So those of you who are extremely industrious overachievers, if you bring me the answer to this question, as well as a full one-page synopsis, just kidding, April Fools, no, just bring me to lecture, come and tell me which tRNA would bind, or which aminoacyl-tRNA, what amino acid would it be carrying, and what would it be called when it's knighted, right? It would carry the amino acid. So for example, arginyl tRNA ARG is currently bound in the P site. So I want you to tell me which aminoacyl tRNA would then bind at the A site here. Patrick is going to be totally jazzed because at this point we need to turn our attention to just a little bit of coverage about how gene expression, how it differs in eukaryotes as versus prokaryotes, or bacteria and archaea, more accurately stated. So take a minute to think about some of the inherent differences between eukaryotic cell structure and prokaryotic cell structure. And of course in that you will have some of the nuances of the details of how they must differ. However, one thing that needs to be overtly stated is that rather than having a single RNA polymerase enzyme, remember the epically awesome alpha, beta, beta prime, sigma, omega of the bacterial RNA polymerase. In eukaryotes, there's more than one RNA polymerase, and they have more subunits, more polypeptide chains that comprise them. They are not very creatively named, so in fact they are RNA polymerase 1, RNA polymerase 2, and RNA polymerase 3, but what they do is a little bit different. So RNA, and I'm going to abbreviate, a lot of times people do that, RNA pol 1, RNA pol 2, and I'm going to start with this one, RNA polymerase 2, is going to recognize genes that encode for mRNAs. So it creates the mRNA transcript, whereas RNA pol 1 is going to recognize genes that will be encoding for ribosomal RNAs. And then lastly, RNA pol 3, as you might guess, is going to be tRNAs and other small nuclear RNAs and whatnot, so tRNAs, to broadly describe the role of these 3 different RNA polymerase enzymes, and then to broadly state that they are in fact more complex than what we see in the prokaryotic or bacterial and archaeal world. I also want to make note that promoter sequences differ in eukaryotes. We broadly talked about the sigma 70 recognized TT, GACA, and TODAT, right? The quintessential bacterial promoter. But in eukaryotes, we tend to see instead that there is still a Tata box, so that stays constant. Brett will be happy to know. So there still is a Tata box, but the Tata box occurs at -30 instead of -10. It's generally centered somewhere 30 nucleotides upstream of the +1 transcription start site. There's also a GC box that's at -50, centers around -50 somewhere, and then there's a cat box. Cat litter box, like kind of like that, only different, at -100. So a little bit different with regards to reoccurring promoter sequences. There are some other differences that I want to briefly highlight, and this is something that we can think about why they might be important differences. For example, eukaryotic mRNAs are going to be more likely to be modified after transcription, and they're more heavily fortified than bacterial mRNAs. We can think about why that is as we write down the first fortification. The first fortification is the 5' cap. So the 5' cap is actually 7-methylguanosine, so it's a specific methylated guanosine residue. I'll just jot that down for those with a sexy mind who want to understand what the 5' cap actually is. And having this on the 5' end of the mRNA is very protective, and it's also thought to enhance translation. So it's a fortification and an enhancer of subsequent processes. So that is one thing that is very different. Additionally, there's a poly-A tail. So polyadenylation, you can see how adding up to 200 adenine residues on the 3' end of an mRNA could really enable it to live longer, so to speak, without getting degraded by those Pac-Man nucleases that chew it from the 3' end. So a lot, up to 200 adenine residues are added onto the 3' end and really enhance longevity of that transcript. And we can think about why that would be so very important. Again, thinking back to compartmentation, that's like the reoccurring theme, compartmentation. Because remember that the mRNA has to travel from the nucleus where transcription takes place out into the cytosol where translation takes place. And so it's got this big journey, an epic journey that it has to go through in between transcription and translation. And so having these awesome fortifications really enables the transcript to have greater durability in that epic long adventure. You could think about it as being the mRNA armor. The 7-methylguanosine, the 5' cap, and the poly-A tail is like the eukaryotic mRNA armor that protects it in that epic journey that it has to make from the nucleus out into the cytosol. So these are really important, and one note, the 5' cap does not occur in, at least in known bacterial and archaeal systems. Polyadenylation rarely, if ever, occurs in bacteria and archaea, so it is unique, and particularly the 5' cap, unique to eukaryotes. So they help to stabilize and enhance expression of the transcript. They are the armor of the eukaryotic mRNA. Love it. Now, we also mentioned this idea of introns and exons. So in eukaryotic systems, in the DNA, there tends to be this non-coding region. It used to be that people would actually literally refer to that non-coding region as junk DNA. That term has become very passé, and that is so 90s, because we now understand that in fact that region often enhances transcription. So it actually will play a large role. Sorry, I forgot this little section here, make sure to add that on. But we know that that junk DNA that it used to be called, that's no longer a very adequate term for something that does play a role, a functional role. So let's go ahead and write down that introns and exons are expressly referring to coding in the case of exons and non-coding regions in the case of introns. Despite the fact that these introns are non-coding regions, we know that they may play a role in enhancing transcription and are not, in fact, junk DNA at all. But it is of note to look at how the mRNA would first be created and how it would have to be modified and acted upon in order to remove the introns and to splice together the exons to create fully functional and mature mRNA. So this is the initial precursor mRNA that includes all the coding and non-coding regions. So when the DNA is first read to make RNA, everything goes. But then the introns need to be taken out. And so, interestingly enough, there are little consensus sequences on to the sides of the introns that allow for recognition. And then there is an incredibly adept machinery, and the name of this just never ceases to strike my fancy. The small nuclear ribonucleoproteins. Small nuclear ribonucleoproteins. What a mouthful! And we don't ever call them by that name. Instead, we call them lovingly SNRPs. So SNRPs are going to cleave out the introns and splice together the exons. And this is an incredibly elaborate process done by something that sounds like a cartoon we may have watched as children. SNRPs come in and splice out the introns and splice together the exons. And so we will write that down. Precursor mRNA becomes mature mRNA when the introns are removed and the exons are spliced together. And now this mature mRNA can be very adequately read by a ribosome and made into its resulting protein product, since the SNRPs worked on it first to splice out the introns and splice together the exons. We're going to stop there for today, and we can be so jazzed to talk about gene regulation next time. So we will get into the nuances of things like DNA binding proteins that can get in the way of expression of a gene. So this is going to be a ragingly good time.